One of the things that we'll probably end up talking about, because you've studied it specifically, has to do with juvenile play behavior in rats, which is sexually dimorphic, and we'll talk about that. But before we get to the rat stuff specifically, can you just talk about juvenile play behavior in mammals generally? How common is it, and what do we think the general biological function it serves as?
Yeah. So thanks for that question. So first, a little background on play. In that it's juvenile play behavior, as you said, and so it only occurs during a restricted period of time, and it's usually in our rodents. It's like just around weaning and until puberty. And once you get past puberty, interest in play goes away, and instead you're interested in mating and being territorial and etc. And it's a mystery still to this day what the purpose of play is. Somehow we all think intuitively it must be important, right? But nobody, and it's debated by psychologists, psychoanalysts, parents, teachers, and neuroscientists. And it used to be a very intense topic of investigation by neuroscientists back in the 1980s, because back then the rat was the favored animal model for neuroscience. Then the transgenics, the power of the transgenics mouse swept in the 90s. And of course, that just, really pushed aside most of the research in rats because it was so powerful to have this ability to manipulate the genome. And it turns out mice are actually one of the very, very few species that do not engage in this very complex play behavior. So what do I mean by complex play behavior? When you put two rats together, so just like puppies or kittens, they will chase each other. They will, one will pounce on the other, and the other one will roll over onto its back to expose its ventrum. And then they'll jump up and they'll reverse, and the other one will chase the other one, and they'll do the same things. They'll stand up and they'll box with each other. And sometimes they just wrestle, they just roll around and wrestle. And we know it's rewarding. They like it. If you let them play in a place that they condition to it, and they want to come back to that place. There's no aggression. There's never any biting. It's when it's going well, it's reciprocal. If you get into a situation where one animal is always dominating the other and sort of pinning it, the play extinguishes because the other one's like, you're not any fun. You don't let me pin you, kind of thing. And that's also true, in so many species that we know of, that we all of our pets and everything. One of the fascinating things I think about our pets is that they are, particularly dogs, they still play post puberty, which is really another interesting question. The reason that I got so interested in it is that across all species that play, males will play more intensely, more physically, more frequently than females. Females do play. They are very physical. They're just not quite as intense and physical as males. And what fascinates me about that is that when they are at the age that they're playing, there's no steroid hormones around, right? This is like, think about like a 6 or 7-year-old child, right? They don't have any hormones at this time, right? So the sex difference is coded by the hormones early in development, just like adult sex behavior and adult aggression, but it's expressed at the time that there's no hormones around. And so that means you're really comparing apples to apples, right? As opposed to in adulthood when males and females have very different hormonal profiles. And so I also think that the fact that there's this difference between males and females can maybe open a window into what is the purpose of play. Do we really need play, kind of thing? And that's one of the avenues that my lab is exploring right now.
Mm-hmm. One way I suppose you could look at that, and I don't know if this has been done, has anyone simply tried depriving juvenile rats, say, of play behavior and see what the downstream consequences are for their reproductive behavior or something like that?
What an excellent idea. Yes. That was done. It has been done many times back in the 80s, but mostly it was done by just completely isolating the animals. So putting them alone in a cage away from all other animals. And it turns out that's about one of the meanest things you can do to a juvenile rat. They're very, very social animals. And it's very, very stressful for them to be isolated. So what we've been doing in my lab, and I should also say it was all only done in males, because all of the research back in the early days was only done in male animals. So one of the things that we've done, a graduate student named Ashley Marquardt in the laboratory, is she created a cage, a normal rat cage, in which she put a perforated barrier, plexiglass perforated barrier down the middle. so that you can put an animal on each side and they can see each other, they can hear each other, they can smell each other, they can tiny little bit touch each other, but they can't play. And so she houses them like that through the entire juvenile period. And then she gets them through, takes them back out, puts them back into group housing at puberty. And then they go into, and in adulthood, she's measured a whole lot of behaviors. And what she has found is that the males show impaired sexual behavior. They're slower to interact with the female sexually, and they interact less frequently. They show a longer latency. They have fewer mounts, fewer ejaculations, etc. Conversely, they seem to be hyper aggressive. So sometimes people say, oh, play is a rehearsal for aggression. No, play is probably a rehearsal for appropriate aggression, not, and so they're much quicker to attack a stranger, etc. They have a very bizarre response to sort of a social preference test, and then would say also it's like a hyper exaggerated response, which we think has to do with dominance hierarchies and not knowing how to interact to establish dominance hierarchies. And then they show a little bit of cognitive impairment. Fascinatingly, the females are completely normal. They show no effect whatsoever of having been deprived of play. And further, if in the middle of this period of this, not being allowed to play, if we free them from these barrier cages and allow them to have a play date, the males are exuberantly playful, right? I mean, they just show a huge exaggeration in their play. And the females are like, they just are, again, unaffected. So it's a really fascinating difference. That why would the play be important to one sex and not the other? And again, it's just, we're just really fascinated by why is it the females, are they just resilient to the stress? When they sit in the cage together, I keep going like this because they sit right next to each other. They know the other animals across the barrier, right? And they sit right there. And somehow that's just enough socialization for the females. I don't know, maybe they're chatting away or something.
Well, yeah, I mean, that was another question I had is when we talk about this play behavior in rats, we're referring typically to this easy to measure, easy to see, like rough and tumble wrestling type behavior. Conceivably, there's other ways they interact that are more difficult to discern.
Yes, that's an excellent point. And in fact, the play is so fast and vigorous, we have to videotape it and then slow it down to score it. And so one of the exciting things that's happening in neuroscience right now is computer vision learning. So you can use artificial intelligence and so we started this process. There's various programs out there. There's quite a few of them that are developing to see if the computer can see something that we're not seeing, right? But it's the challenge is that these two animals, you can teach the computer to track one, and we can get the computer to follow them around as they're like chasing each other. But as soon as then they interact and then they become a furry ball, right? And then it's really hard for the computer to tell who's who. So we're working on that. But you raise an excellent point. Is that we do think that there could be things that are going on that we aren't detecting. There could be ultrasonic vocalizations that they're making to communicate with each other that we can't discern. And again, these are just technical problems because we can record ultrasonic vocalizations, but we can't tell which one is vocalizing, right? Because so things of that sort. So, but I think those are exciting questions to pursue in the future.
Yeah, well, you said a couple of things that were super interesting. So when we think about depriving the rats of play, there was this dimorphism there. So the deprivation of play affected males more than females. It resulted in subsequent deficits in their adult behavior, such as the reproductive behavior and their propensity towards aggression, as you mentioned, which is maybe a little bit counterintuitive to people, but I think not so much once you think about it. If you don't let the male rats do rough and tumble play with each other, then they become hyper aggressive. They basically don't know how to appropriately interact with other males and other members of the species later on. And you also said that if you deprive them for a little while, then they become like really, really excited to play even more. So that implies that this is almost homeostatically regulated the same way that other processes in the body are, which would also say that something very crucial is happening here.
Yeah, that's interesting. I'm glad you said that. I hadn't thought about it in terms of being homeostatically regulated, but you're absolutely right. In fact, we know sometimes in our experiments, if we want to do things like we want to look at the neurons that are activated by a play bout, right? So we'll look at an immediate early genes expression in a particular brain region after a play date. What we'll do is we'll isolate the animals for 24 hours before the play date. And then we know we'll get this huge exaggeration in play. And so you're right. It's like, or other times what we do is we let them play together every day for like 10 days in a row. They meet a playmate in the afternoon that they go, and it's not their cage mate, it's not their sibling, it's their playmate. And they go to a neutral arena, it's not in their home cage, and they get about 15 minutes to play. And they'll just steadily play the same amount pretty much across the whole 10 days. Kind of thing. So unless we isolate them, and then boom. And if we introduce a new playmate, that it doesn't affect the males, but the females, they're like, I don't know who you are, and they don't play with the introduced animal.
And now, will juvenile male rats preferentially play with other males, or do they play differently with juvenile males versus females?
Yeah, that's a great question. If we look in terms of the intensity of play, the highest is male to male. Medium is male to female, and lowest is female to female. And when you do mixed groups, because what we do now is we started out, we would do like 6 animals together, and we would just watch the all 6 animals, which was crazy. Yeah, because you got to label them and all that kind of stuff. It's a good question whether or not the males initiated more with other males. And that's where, like, if we could get really sophisticated computer learning would be vision learning would be a good question to ask. But definitely, one of the challenges of studying the behavior is that it does require reciprocity. And so if the other animal is not playing the same, that will kind of extinguish it. And so we've been looking at things like rats with genetic mutations for genes that are identified in autism, and to see if their play behavior has changed. And it is, and it's reduced. And what we have to do is we have to put the two, like the genetically mutated rats, two together. Like, we can't do a mixed dyad because it won't reveal the effects. But so you have to sort of exaggerate it. But anyway, I whether or not there's a preference for a male with a male, I don't think so. But they'll have more reciprocity from a male, and so that just naturally exaggerates the behavior.
I see. So this play behavior is sexually dimorphic, both in terms of how vigorously and how frequently it happens in males versus females. It's also dimorphic in terms of how important or influential it is on their subsequent adult behavior. Because if you deprive males, they have deficits in reproduction and aggressive behavior, as you said. You don't see that in females. So the next question is really, okay, this is super interesting. Where does it come from? And I know that a lot of your work has looked at how this type of difference develops and how the endocannabinoid system in particular ties into that. So can you start to tell us that story of where this dimorphism comes from?
Yeah. So first you have to start is, what is the neural circuitry of play, right? In the brain in general, where does play come from? And there is a very strong consensus in the field now of what we call the social behavior network. And now there's also what's called a core aggression network. And these are brain regions that are interconnected that regulate social behaviors, and then also aggressive behavior. And they usually start with the olfactory system, either the main or accessory olfactory system. And then you're going to have projections to the amygdala. You're going to have participation of the prefrontal cortex. You're going to have a component of the reward pathway, the VTA nucleus accumbens, the BNST, and then you sort of kind of go down to the midbrain to the spinal cord. The neural circuitry of play, near as we can tell, we don't know it as well as we do the others, because a lot of that has been really finely mapped now in the mouse. The neural circuitry of play is overlapping with the social behavior network. It's the same network, which is also fascinating, because then, like, why does play go away, right? Why does it extinguish with puberty? But fascinatingly, the sex difference is only in the medial amygdala. You could imagine it being a distributed property, right? That would it would be redundant through the entire social behavior network, but it's not. It's only in the medial amygdala. And why do we know that? We know that because if you take a newborn female rat pup and you put testosterone only in her medial amygdala, 3 weeks later or 4 weeks later, when she starts to play, she'll play like a male, right? You don't have to have the testosterone anywhere else in the brain. So we know that.
So there's one spot medial amygdala in this case, you put testosterone in a female, so it's at levels that resemble what would be naturally in a male. And later on, when she becomes a juvenile, she'll play like a male.
Right. Right. So my question was, what is that testosterone doing? Right. What's its cellular mechanism of action? And we had been doing some other studies on the hippocampus where we had seen some sex differences in neurogenesis, actually with males making more new neurons than females in the first couple of days of life. And I had a new postdoc in the lab named Desiree Krebs-Kraft. And I said, well, the amygdala is in the same sections as the hippocampus. Why don't you just go and look at the newborn cells in the amygdala and see if there's any sex difference there? So she did. We do this with a BRDU injection. That's a thymidine analog that labels newborn cells. You have to then section the brain and look at it. And she went and counted the newborn cells in the medial amygdala. And lo and behold, she found that there were more newborn cells in the female than in the male. And that was exciting because, usually everything's always more in the male, more in the male, more in the male. And so she's like, oh, well, that's cool. So there's more newborn cells in the female than the male. And then she's like, well, now what? I was like, yeah, now what? That's a good question. Well, sometimes science occurs in funny ways. I had a colleague named Brad Alger who studied the endocannabinoid system. He was one of the pioneers of the system. And after, at this point, I think we've been colleagues for 20 years or something. And every single seminar, every thesis defense, every journal club, every thesis proposal during the question and answer session, he would stand up and say, endocannabinoids can explain that, whatever it was, whatever phenomena it was, literally. So I just literally said to her, I don't know, let's look at endocannabinoids. Maybe they're regulating that's literally how it happened. And so she said, okay. And she took a pan agonist for the CB1 and CB2 receptor, which are the two receptors on which endocannabinoids and also THC act. And she injected animals. We just did a pharmacology experiment, and she was able to reverse the sex difference. She was able to bring down the number of newborn cells in the female to the level of the male. So she was able to eliminate the sex difference. And since we knew from the previous work in the 1980s of Bruce McEwen and Michael Meany that the medial amygdala was critical to the sex difference and play behavior, she then looked at social play behavior, and she found that it also, the treatment with the pharmaceutical drugs that mimicked THC had also sex reversed the play behavior. And that set us off on this journey to understand really precisely what the endocannabinoids were doing.
So just to reiterate, so you give testosterone to a female, specifically in the medial amygdala. Her subsequent play as a juvenile will look like a male. And the same thing happens if you give an activator of CB1, CB2?
Correct. And you don't even have to give it into the amygdala. We just give it under the skin. We give it sub-Q.
I see. Yeah. And does that happen also with THC itself?
Yes, it does. We haven't published that work yet. So yes, but we have, and this is Jonathan Van Rissen, who's been carrying on this work, and he's preparing the manuscript now, in which if we inject newborn pups with THC at exactly the same time that we would do testosterone, it has the same effect on the newborn cells, and it has the same effect on the play behavior. Interestingly, if he gives THC to pregnant rats earlier, so we're looking at, because we're trying to model human cannabis use during pregnancy, if we give it much earlier, it has the opposite effect in terms of play behavior. It actually suppresses play behavior. So it promotes play behavior in the young animals when you give it to the newborns, and it suppresses it if it's given during fetal life. And we don't know the mechanism of that. But again, it's just the importance of timing, development. Mm-hmm. It's all about timing.
Yeah, you can get the opposite effect at two different times with the same treatment.
Right.
So is there some kind of interaction you guys are hunting down between the sex hormones like testosterone and the endocannabinoid receptors? Do we know exactly where that link is?
Right. So this is where the story got really, really interesting. We did all those obvious things, right? So we looked for sex differences in the endocannabinoid receptors and sex differences in the endocannabinoids themselves. And we did find that, in fact, males have a higher level of the endogenous endocannabinoid called 2-AG. And endocannabinoids are a funny signaling system, right? They are a membrane-derived signaling molecule that that's sort of continuously produced all the time. And so there's like a tone, right? You have like a certain tone of endocannabinoids that you sit at. And so the conclusion was that males have a higher endocannabinoid tone than females. And if we give females testosterone, they make more, they they have a higher tone. It's either because they're making more or they're degrading it less. A big part of the endocannabinoid system is, it's like, think about it as well, how to think about it. But basically, you make it and you degrade it, and you can either make it faster or degrade it faster, and that will shift the amount either way. So it's definitely the endocannabinoid tone, not the receptors, that was different. But then it was like, well, what are the endocannabinoids actually doing? And we were thinking about the adult and all the things that are known in the adult about how it regulates synaptic physiology. They have, presynaptic inhibition, depolarization, induced suppression, et cetera. And none of that applied to the development of the amygdala, which it makes sense in retrospect because it's just not developed yet. It doesn't have all that synaptic activity. And so we're trying to think about cell genesis. So the big question was, who are those newborn cells, right? There's born. And of course, you think it's going to be neurons. And certainly in the hippocampus, we had shown it was neurons. But again, in retrospect, it makes sense. They weren't neurons at all. It was the glial cells known as the astrocytes, right? So these are the sort of, had always been considered the support cells of the brain. But we now know they're actually very, very active cells, and that they're really important to synaptic physiology and et cetera. And Desiree first identified that, in fact, that it did look like it was the astrocytes, that there was more in the females than the males, Desiree Krebs-Kraft. And then Jonathan Van Risen really confirmed that, but he added in the really interesting and unexpected twist of that he brought in a third cell type called microglia. Now, the problem with microglia is their name. They are not glia. They are not astrocytes in any way. And they're not micro either, because they're 10% of your brain. What they actually are a modified immune cell. And it's a modified macrophage. And they are born very early in development, and they get into your brain and they take up residence and they stay there your whole life. And they're scattered throughout, they're peppered. Actually, we call it, they're tiled through your entire brain. Your entire brain is full of microglia, and they're there as your brain's immune system. They're unique to the brain. They're nowhere else in the body. And what Jonathan found was that rather than, it used to be, we thought microglia, we called them quiescent. We thought they just sat there waiting for an accident to happen. And then they would maybe, release cytokines or prostaglandins and respond to the injury. With the advent of the transgenic mice and the amazing things that you can do with transgenic mice, we were able to visualize them in the living brain and found out that they're not quiescent at all. They're actually moving around all the time, and they're always checking on the other cells around them. But what Jonathan found, and again, just to repeat, Jonathan Van Risen, is that these macrophages, these immune cells, were in fact engulfing the newborn cells in large phagocytic cups, the living newborn cells, and they were actually then killing and consuming them. Basically, by consuming, I mean they degrade them.
So these are effectively like immune cells of the brain. And just like a white blood cell or something in your body might eat a bacteria from an infection, they're eating newborn neurons in this case.
Newborn astrocytes.
Newborn astrocytes. Okay.
Yeah. Yes. That's exactly right. And in an exquisitely tightly controlled manner, which is what's also fascinating. And that control is coming from the endocannabinoid tone, right? So the higher endocannabinoid tone in the males acts on the microglia to make them basically give them the munchies. Right? It makes them hungrier, and they consume more of the newborn astrocytes. And so the females wind up with more of those astrocytes surviving. And so that as they get to the time of the juvenile playtime, the female's medial amygdala has more resident astrocytes than the male does. And you still say, well, what does that have to do with play? Well, what Jonathan Van Risen is showing now is that those astrocytes are actually inhibiting the play neurons because they're releasing adenosine, a chemical called adenosine. So the more astrocytes you have, the more inhibition you have of the play neurons. And the males have escaped from that break, basically, by killing off their astrocytes when they were young.
I see. I don't know if I want to go on this tangent, but you said adenosine is involved here. That immediately makes me think about caffeine.
Oh. Wow. Yeah. I don't know.
Okay. So so basically development is happening in males and females, naturally under normal conditions, you'll have different levels of endocannabinoids. They'll be higher in males, I think, is what I'm hearing. And that will make the microglia hungrier, and they'll literally eat and digest these things called astrocytes. And that sort of releases the ability to have more play behavior in juvenile males and suppresses it in females.
Exactly. And interestingly, they only eat the newborn astrocytes, right? They like the babies, right? Which is, once the astrocytes are mature, they don't seem to consume them.
So a natural question here is, similar to what I asked before about deprivation of play. You said that if you deprive a male of its ability to do rough and tumble play with other juveniles, it has downstream consequences, negative consequences on the adult behavior, both on the aggression side and the reproduction side. What happens to adult behavior if you say masculinize the females or do the reverse? Does something similar happen downstream?
Yeah, that's an excellent question, and we have not done that experiment. That's because I think that, so if we gave hormones to do that experiment, it like to peripherally say, then it would masculinize everything, right? Because it would invoke another mechanism in the hypothalamus and another mechanism in the hippocampus, etc. If we give the endocannabinoids, it's interesting. What we have to do. So when I give a newborn female testosterone and I turn her brain male, and if I take her to adulthood and I want to see her do male sex behavior, I have to give her testosterone again, right? Because even a normal male will not show sex behavior without testosterone.
I see.
But you raised an interesting idea. I have not thought about if we gave the endocannabinoids, could we get them with just adult testosterone to start to show male-like sex behavior? That's an interesting idea. I got to think about that. Potential prediction would be that, if you're masculinizing the play behavior and the juvenile play behavior is causally related to adult reproductive behavior, would at least in the presence of testosterone, would a masculinized female start exhibiting male-like mating behavior as an adult? Great idea. I like that. I think we'll try it.